Hi everyone, welcome to your video on mitosis. This is the very first lesson in cycle 3, which is titled change. So our whole theme for this cycle will revolve around the word change. So just a few things to remember. Cells are made up of organelles, and you guys did an in-depth study of all the different organelles in plant and both animal cells. Cells take in and release energy and materials. In some jobs, some cells are specialized, which means they only perform certain jobs. So cells have to grow and divide. Cell division is going to be the topic of this lesson, and it corresponds to our theme of change because cells are constantly changing and growing and dividing. Cell division occurs in all organisms. Unicellular organisms reproduce through cell division, and multicellular organisms need cell division for growth, development, and repair of their cells. So right now, we're multicellular organisms, humans are, and right now our cells are growing, developing, and repairing themselves. And you are growing because your cells are dividing, which makes new cells, and that makes you grow. So there's something called the cell cycle, and in order for new cells to be made, cells have to go through the cell cycle, similar to our life cycle. And this only happens in eukaryotic cells. Remember that prokaryotes don't have a nucleus, and the cell cycle has to deal with the nucleus and starts with the DNA in our nucleus. So the cell cycle consists of two main phases. It consists of interphase, and then it consists of mitosis and cytokinesis. Cyto, you think of cytoplasm, and kinesis. The word kinesis refers to movement. So the cytoplasm is moving, and we'll get to what that is later, but I just wanted to help pick apart that big word for you. Okay, so we're going to start with interphase. The part of the cell cycle in which the cell is growing and is not dividing. So it's just the cell kind of hanging out and growing. In addition to growing, the cell's carrying out its normal activities. It's taking in materials, it's getting rid of waste. Inside the nucleus, the cell's DNA is being copied. So there's 2 copies of each chromosome are made before mitosis even starts. That is all part of interphase, which again is not actually part of the mitosis phases itself. Okay, before we get into it, we need to identify a few key terms that we're going to be using throughout this lesson. DNA. DNA is a chemical that contains information for an organism's growth and functions. Deoxyribonucleic acid is how you say that. Deoxyribonucleic acid, DNA. In eukaryotic cells, the DNA is found in the nucleus, and in prokaryotes, it's in the cytoplasm. Remember, prokaryotes don't have a nucleus, and the DNA just floats around in the cytoplasm instead of inside the nucleus itself. And we're going to get into DNA a little bit more. You're going to do a really cool web quest on DNA. And you see this picture that's showing you base pairs. These base pairs are always together, and you'll learn why and how different structures are formed a little bit later. Okay, so DNA is wrapped around proteins like thread around a spool and compacted into structures called chromosomes. So chromosomes are going to be the basis of mitosis, of each phase of mitosis. They're going to be changing each time. Here is a picture of chromosomes. The top one is showing you the nucleus, showing you a cell, and inside the cell is the nucleus, and then the chromosomes live inside the nucleus. And the bottom one shows you that the centromere is the middle part of the chromosome that holds it together and gives it the X shape. And then you see the little blue dots. Those are kind of like the chromosome's knees. And those are actually genes. Those are segments of DNA that codes for certain traits. So you have specific genes from your parents. That's where they actually live on your chromosomes. And then at the very bottom, like the feet of the chromosome, those are the chromatids or chromatids. It's said differently depending on who's saying it. Okay, so some terms to remember. The centrioles, those are little tubes made up of microtubules, and those are protein fibers or strands. If you did the animal cell model, you may have come across centriole in your research. The nuclear envelope is nothing more than just a little envelope that surrounds the nucleus, helps protect it. Chromatin is unwound DNA. It looks like spaghetti. Chromatid or chromatid is the legs of the chromosome, which I just showed you in the previous picture. And the nucleolus is the little blob inside the nucleus where the ribosomes are made for the cell. Okay, so here's a picture of everything that we just went over. So you see the nuclear envelope is the line around the nucleus. The nucleolus you see is that blob inside of the nucleus. Chromatin you can see as the purple inside of the nucleus. Centrioles you can see on the outside. Those are the yellow dots outside of the nucleus. Okay, so here's the first diagram that you're going to see relating to mitosis. So if you notice it, just start up at the top left and you see prophase, then moving over you see metaphase, then moving over one more you see anaphase, and then at the bottom you see telophase, and then moving over you see interphase. So by looking at this, it looks like you actually end with interphase, but remember this is a cycle, so you start and end with it. So I'm going to come back to this diagram each time I go over a phase, and the first phase that we're going to go over is prophase. So prophase is the first stage. This is where the centrioles move towards opposite ends of the cell, which the cell is shaped sort of like the Earth. So we're going to refer to opposite ends as poles, just like the North and South Pole of Earth. So as they separate, spindle fibers, which come from the centrioles, these are kind of like spider webs coming out of Spider-Man's hand. These spindle fibers between them grow and keep them connected, sort of like if there were a string between them. The nucleolus and the nuclear envelope disappear, and then the chromatin coils up and makes chromosomes. They couldn't get out if the nucleolus and the nuclear envelope didn't disappear. So let's look at the diagram. We have a couple different diagrams, and if you look at the top diagram, you will see the centrioles that are at the top and the bottom at the poles. You'll see the stringy-like things connecting the poles. Those are the spindle fibers that came out of the centrioles. You'll see the white dashed circle breaking apart. That's the nuclear envelope. And then you'll see the chromosomes starting to get out. Each one of those is very similar. They're all representations of prophase. I prefer the top left beigeish one with the blue because it matches most closely what you just wrote down. All right, going back to prophase, you see the centriole. In this one, they're red. You see the purple blob in the middle is the nucleus. The nuclear envelope is disappearing, and you see the chromosomes and the chromatids in the middle. All right, next we're going to move on to metaphase. So metaphase means meet in the middle. Chromosomes are pulled by spindle fibers, those little spider webby things, so they line up in the middle of the cell. So now you can see the chromosomes, which are the X's, lined up completely along the middle, kind of like at the equator. The centrioles are at the North and South Pole with the chromosomes lined up along the equator. This is what metaphase is. Again, look at metaphase. We're at the second diagram over. You see the centrioles at the top and the bottom, and the X's along the middle. Meta, middle. The next one is anaphase. Anaphase, so each sister chromosome gets separated. So our X's, our X chromosomes, pull apart and turn into V's, and they go to opposite sides of the pole, or opposite poles of the cell, rather. So you can see on the right diagram, either of those diagrams, you can see that they were once X's that are now pulled apart into little V's, or U's, depending on which way you look at it. The chromosomes separate by tearing apart at the centromere. The centromere is the middle of the X that holds it together. Okay, so if you look at the top right, that's anaphase. The X's were split into half, basically, just split into two. and they move to each pole. The next one is telophase. Telophase, the chromosomes move in sets at the 2 poles. The chromosomes begin to uncoil back to chromatin, and look like they did during interphase. Okay, so they're getting back. Remember, this is a cycle, so they're starting to get back to what they looked like during interphase, where the cell is growing. The nuclear envelope reforms around each chromosome set, the spindles disappear, and the nucleolus reforms. Nuclear division by mitosis is complete at this point. So if you look at the bottom left, you see telophase. We have now formed, we're in the process of forming 2 cells. So you start with 1 up at prophase, and then down at telophase, we're moving into 2. And you can see where it's pinching together in the middle in telophase. Obviously, you know something happens because if you look over to interphase, you have 2 different cells. So there's one more thing we need to talk about. And that is cytokinesis. So the cell curves in and splits into 2 identical, they're called daughter cells. Each one of those goes into interphase. So you now have 2 cells going into interphase that are going to grow, and then they will each start the process of mitosis over, and that's how they will grow and divide. And this differs slightly in plant and animal cells. And I want to just say, it's okay. If you're freaking out because this seems like a lot of new material, it is, and you will have lots of practice. So don't stress out. I do want to remind you to please read over your graphic organizers. Read over them daily. Don't just look at this when you're doing the video. You have to practice the stuff and see the words and hear them in order for you to really understand it. And we'll be doing lots of practice with this in class. Awesome. Have a great